Hey there, pre-med. In this video, we're going to talk about the neuron, which is a very important specialized cell in our body, and how the neuron conducts neural impulses through action potentials. We're going to focus on the cellular structure and the action potential today, but of course it's important to put this in context of our overall nervous system, which allows us to take in signals from our environment and respond to them. So if I grab a cup of water, and I'm like, oh, this is really cold, right? And I might want to change my placement of my hand on this cup to the top because it's really cold right here with the water. That's a signal, a neural impulse that allows me to feel the cold and respond by changing my hand position. So today we're going to focus on what's happening in individual neuron cells to allow that transmission of signaling and see how there's actually a physics connection in this biology topic. Before we dive into the content, we're going to check out an MCAT style practice question to give us some context of the depth that this topic is tested on our exam. Let's get started. Go ahead and read through this question, give it a try on your own first, and then we'll come back together, talk through the different terms in this MCAT style question, and review the underlying content. Go ahead and give it a try. The question reads, the resting membrane potential of a neuron depends on Roman numeral options. Roman numeral 1, the opening of voltage-gated sodium channels. Roman numeral number 2, the active transport of sodium and potassium ions. And Roman numeral 3, the passive diffusion through potassium leak channels. We can see here that we have several options. I always like to look, before I even try to attempt a Roman numeral style question, I do look and see if any of the answer options are in all four answers. Because if any of those answer options are in all four answers, we don't even have to worry about it. It's a correct option. Unfortunately here, none of these are in all four, right? A, B, C, or D. And we can see that there's different patterns. We have 1 only, 1, 2, and 3, 2 and 3 only, and 1 and 2 only. So we're really going to need to think through the content here. So we're going to take a step back and talk through the content before diving back into this question. In order to understand the resting potential of a neuron, we first need to talk about what a neuron is. I want you to notice a word here that should ping your brain from your physics topics, which is potential. Resting potential. Doesn't that sound kind of like a 4C electrostatic circuits voltage topic? And we'll see how this potential is referring to a voltage. It's referring to a voltage or a difference in charge across a membrane. So we're going to talk through the difference in charges for the neuron and how these different ions help to maintain specific ranges of voltage. Let's first dive in with the anatomy of a neuron so we understand where these things are happening. We have here a diagram of a neuron, and it's actually more accurate than it looks. It looks like a cartoon, but it's actually pretty accurate. And we're going to go through and we'll first annotate and describe each part of the neuron. On the left here, we have these kind of tree branching structures, and these are known as dendrites. Dendrites are the part of the neuron that receive a signal. So they'll either receive it from other neurons or from surrounding cells. These signals usually come in the form of neurotransmitters. Neurotransmitters are the signals that we use between neurons or between neurons and other cells. So we'll use them right now as a general term, neurotransmitters. Another video where we're going to talk about the neuromuscular junction, we're going to get more into the different types of neurotransmitters. So the dendrites, these little kind of tree-like, branch-like projections, receive signals. And we have here the cell body, or the soma. This is where all of our normal cell structures that we expect to see are, okay? This is where our nucleus is, our mitochondria. So all our normal cell features that we expect are in our cell body or our soma. Like I said, the neuron's a very specialized differentiated cell, so it's got a very unique structure. Moving forward, we have this long piece, and this long piece can be really long, and this is called the axon. The axon is where the signal travels. So when we want to send the signal off to a new cell or another neuron, we will send the signal from our dendritic side, from our cell body side, down the axon and to what we call our synaptic terminal or nerve terminal. So in this axon, we've got a couple of key features. The first is the point at which the axon kind of connects to the more cell body dendritic side of the neuron is called the axon hillock. And this is very important because this is where our signal originates to go down the axon. So I kind of think about it as like, here's where the signal forms. And I like the word hillock because we're going to talk about this kind of all or nothing threshold-based action potential. And I like to think about it as the threshold is like we're at the top of a hill, and once that signal gets over a certain threshold, we're rolling that ball down the hill, and that down the hill is kind of like almost like a roller coaster, and that's through our axon. So I like the word hillock because it kind of has that visual of reaching a threshold and then swinging all the way down through the axon. Now the axon can, like I said, it can be really long. In fact, our longest nerve in our body is our sciatic nerve. And this guy can be over 3 feet long, which is wild to me. So just note that these nerves, they're not teeny necessarily. They can be, but they can also be super long. And so a big part of our body efficiency in terms of engineering is how do we make sure that we have this signal and the signal doesn't dissipate, doesn't fade away as it moves through these very long axons. And one of the ways we do that is with these fun little yellow things. These fun little yellow things are called myelin sheaths. Myelin sheaths insulate the axon, which allows the signal to transfer faster. We're going to talk through exactly the mechanism of how that happens in just a second. For right now, you can just write in your notes, myelin sheaths increases the speed of transmission by insulating the axon. Much like if we have a signal like in our power lines, they're insulated, right? They're insulated wires so that that power doesn't dissipate into the environment. So we're going to talk about how exactly that works. And then these fun little purple cells are called Schwann cells. Schwann cells actually create myelin in our peripheral nervous system. So the peripheral nervous system is everything except the brain and spinal cord. So our Schwann cells are the ones that create myelin in our peripheral nervous system. If we're looking at central nervous system cells, those myelin sheaths are created by oligodendrocytes. In my notes, I like to put Schwann cells and oligodendrocytes right next to each other as they're essentially equivalent in function, just different in terms of location. Finally, our last anatomical term for our neuron is these little spaces between the myelin sheaths. I'm going to go ahead and draw a long arrow. These are called the nodes of Ranvier. And these are where the action potentials take place in a myelinated neuron, and the action potential signal kind of jumps from node to node. And we're going to talk about the mechanism of how that happens in just a second. Okay, we have our labeled neuron. Again, most neurons follow this function, although they are very specialized. So we will see slightly different structural forms of like the dendritic part of the cell in particular, how they branch depending on the part of the body we're in. For the MCAT, we just need to know the general terminology and anatomy of a neuron cell. Let's now get into what's happening when we receive a signal, again, from the environment, another cell, another neuron, and then decide to propagate that signal, to continue that signal down through an axon. And the way we do this is with an action potential. Here we have a picture you may recognize from your biology classes. This is our action potential on a time voltage curve. So our voltage is going to be on the y-axis here. All right, technically it's going to be in millivolts because these voltage changes are really small when we're talking about neurons. Okay, so I'm going to draw just a loose picture of an axon. All right, so just imagine our dendrite and somas here. Here's maybe our action hillock. And then we have our axon here. So I'm going to be showing you now at the cellular level what is happening. When our neuron is just chilling, it's not doing anything, it's not sending signals, we are at what's called the resting membrane potential, which you may remember from our practice question. And our resting membrane potential for most neurons is negative 70 millivolts. Okay, let's think about this from a physics section topic. So this is 4C, right, where we're talking about voltage, electric potential, electrostatics. When we're talking about a non-zero voltage, that means that we have a charge differential, a difference in negative and positive charges across a membrane. So what's happening at resting is if we have negative 70, that means inside the axon is more negative than outside. All right, so outside is going to be, relatively speaking, more positive. And so when we have a voltage potential across a membrane, a cell, that is negative, what that means is the interior of that cell is more negative. Now, again, depending on how much we've refreshed our 4C topics, you may note that when we have this differential, we can also draw electric field lines. And electric field lines always point in the direction that a positive charge would go. So a positive charge is going to be attracted to a negative charge, which means our electric field lines, our E field lines, are going to be pointing in to our neuron. So this is something that the MCAT really likes to test because we're combining bio and physics here, is because there's a charge differential, we're going to be pointing perpendicular through the membrane and pointing in because, again, the electric field lines point in a direction a positive charge would move and it would be attracted to that negative charge that's inside the axon. So that's just a fun, quick little physics connection that is often tested on the MCAT, and we'll use this concept of voltage differential to understand what the driving forces are behind these changing voltages that happen once we start our action potential. Now, before we dive into what happens when we start a signal, we have to talk about how we're maintaining this negative 70 millivolts. How are we keeping this negative 70 millivolts nice and steady? And there are 3 ways we do this. I'm going to draw this here on the bottom side, but of course this cell is circular, right? So these channels and pumps that we're going to talk about can be anywhere on the axon membrane. So we have 3 pieces that maintain our resting potential. The first and most commonly thought of when we talk about this topic is our friend, the sodium ion, potassium ion, ATPase pump. All right, so sodium-potassium ATPase is an active transport pump. And so this now is getting into a little of 2A cell biology, where we are talking about the different types of transporters in and out of a cell. So an ATPase uses ATP, uses energy to pump our molecule or ion of interest against its gradient. Okay, so I will tell you and then I'll explain. The sodium is going to be pumped out of the cell. Sodium is a very important ion for us here, so we're going to put sodium in orange, all right? And sodium is going to get pumped out of our neuron because we have much higher sodium concentration outside of the cell relative to inside the cell. It's usually like, I think it's like 12 millimolar inside and then like 150-ish millimolar outside at resting. So you can see, right, there's a big differential here, a gradient. And when we're using a pump, we're pumping from low to high because that goes against our energetics where we always want to go from high to low, right? So we're going to pump sodium out of the cell, right, and into the interstitial space. And we pump 3 sodiums at a time. At the same time, and we're going to put the potassium in bright green here because this is another important ion, we are pumping potassium into the cell. So by definition, because this is a pump, that means our potassium concentration is much higher in the cell than outside the cell, right? So if you know you're pumping and you're pumping in, that means that the higher concentration is inside the cell. This can become real important when we're talking about how these ion levels change. And we pump 2 potassium inside for every 3 that pump out. This is specifically called an antiporter. Antiporters are pumps that pump 2 things, opposite directions, both going against their gradients. So why is this helping us maintain negative 70? Well, look at this. These are both positively charged ions, right? They're both cations, but we're pumping 3 sodium out for every 2 potassium in. So ignoring the actual identity of the ions, 3 positive charges are leaving and only 2 positive charges are entering. So every time this ATPase pumps, we have a net negative 1 charge being developed inside the cell. So this is one of the ways that we maintain this kind of negative 70 is we're pumping more sodium out than potassium in, and that keeps our gradient negative. The second and third components that maintain our resting potential are our leak channels. We have a sodium ion leak channel and a potassium ion leak channel. Now, what are leak channels? Leak channels are truly what they sound. They are these channels that are open and passive, right? So we have passive diffusion, which means the ions are always going to go down their gradient. So potassium in a leak channel is going to go from in to out and leave the cell, right? Leave the cell. Sodium, on the other side, is going to enter the cell because it's going to go with its gradient and be a passive leak channel. Now, both of these are involved. So you're like, how is this actually maintaining this -70, Amanda? This doesn't make sense. It's because we have way more potassium ion leak channels, and we have, relatively speaking, less sodium leak channels. So similar to our 3:2 ratio of our ATPase, we have more potassiums leaving, right, going with their gradient and leaving the cell, and we have less sodium ions entering the cell through the leak channel, which maintains a resting potential that's closer to our equilibrium for potassium. All right, so we're really aggressively separating our sodiums, and our potassiums are a little closer to their natural equilibrium. And we'll see how that's important as we go through the action potential stages. Now, the MCAT can describe these second two pieces in a variety of ways. We can call them leak channels and say that we have a greater abundance of potassium leak channels than sodium. We could also say that there's a difference in the permeability of the membrane to potassium versus sodium, right? Where we have increased permeability for potassium, less permeability for sodium. Or we could say that the passive diffusion rates are different, right? We have a higher passive diffusion rate for potassium and a lower one for sodium. So just note there's a couple different ways we can describe these leak channels, all about permeability and passive diffusion, but they're all describing the same thing, which is saying, hey, more positive charge is leaving the cell than entering, which results in our -70 millivolt balance, and we kind of stay right in that -70 range if we're at rest. All right, we've now set the stage for what happens with a neuron at resting membrane potential. We are really close to answering our MCAT style practice question, but there was one channel type we haven't discussed, and that's the voltage gated channels that allow that action potential to happen. Before we dive into that, I'm Amanda Brem, and I've been coaching pre-med students on their MCAT journeys since 2019. Please remember to subscribe to this channel for more videos on MCAT content, test taking strategies, and mental fitness tips to help you perform your best on test day. And if you'd like more interactive lessons on topics like these, including MCAT style passages and practice questions under timed conditions with me as your instructor, feel free to check out our next available cohort in the caption below. All right, let's dive into that action potential. So the action potential is the signal. It's the electrical signal that travels down the axon and allows us to stimulate neurotransmitters at that synapse and allow that signal to transfer to another neuron, to a muscle, or to another cell, or to just release neurotransmitters to go out and do their thing and interact with other neurons in the body. So how this happens is it's an electrical signal. So we have to really think about the changes in voltage as being the drivers here. In order to reach an action potential and start the process, we need to hit -55 millivolts. Now, if we think about that, that's plus 15 from our -70 resting potential. There's a variety of ways we can get this little bit of positive influx. Most commonly, the neurotransmitters that bound to our dendrites will do a slight depolarization. We'll have a little bit of an influx of sodium ions that allow us to get to that -55. However, it is very important to note anywhere between -70 and -55, if we get to like -60, we're not going to have an action potential. And this is often what's described as the all or nothing response. It doesn't matter if we have a signal or a bunch of signals, if they don't get up to that -55 millivolts in our axon or at our axon hillock, we're not going to get that action potential. Now we can have what's called summation, which is where we add together multiple signals to reach -55. And again, these are depolarizing signals. So when we say depolarizing, we mean getting closer to zero or more positive, so a more positive influx of ions. So if we have these signals that result in us getting more positive charges into the cell, more positive ions into the cell, we can add those together sometimes to reach that -55 and start our action potential. There can also be inhibitory signals though, and those can be summed up as well. Inhibitory signals repolarize the cell. In other words, make the cell more negative, which takes it further away from that -55 threshold. Okay, so that's the different options. We can summate, and as soon as we get that excitatory signal, we are off to the races. Whoop, whoop, we can now have our action potential. And you'll notice it is a very steep increase where we go zooming all the way up to about plus 35 millivolts. All right, so we went from all the way to -55 up to plus 35. And how did that happen? Through our voltage-gated sodium channels. So I have a new axon down here, and our sodium channels that are voltage-gated, I'm gonna write voltage-gated as VG. All right, so our sodium voltage-gated channels are just what they sound like. They are opened and closed by a voltage. And guess what that voltage is? -55. Yep, if you guessed it, you are absolutely right. So what happens is these are closed. These are locked. They're not allowing any ions through until we hit -55, and then that gate pops open. Now, these are passive channels, all right, which means any passive channel means we're going to go down our gradient from high to low. So remind ourselves, where is the higher gradient? Outside the cell. Right? So if we have more sodium outside the cell, what direction is sodium going to go? Into the cell, right? Into the cell. So we're going to have what's called sodium influx. And sodium influx, a bunch of positive charges entering into our cell, is going to, of course, make our cell more positive, which is why we have that increase. Now, not only is the concentration gradient, right, less sodium to more sodium, driving this interaction, but it's also the fact that our cell, when we start off, is negative, right? -55 voltage. So think about that electrostatic attraction, right? Positive charges are attracted to negatives. So relatively speaking, because we have that electrostatic differential, we're also getting an electric driver as well as a concentration gradient. We have an electric gradient and a concentration gradient driving those sodium ions into the cell. So that's what causes that super sharp spike up to plus 35. This is called depolarization, which makes sense as a word, right? Because polar, like further away from neutral. So when we were at rest, we're all the way down at -70. We're now getting closer to 0. So we're depolarizing, and then we just kind of like overshoot a little bit all the way up to plus 35. At plus 35, I'm going to do this in like yellow for us, okay? At plus 35 millivolts, the gates for sodium close. All right, so the gates for sodium close, which means we're no longer getting that influx. And our gates for potassium, our voltage-gated potassium channels, open at plus 35. So not only are we closing our sodium channels at the peak of our action potential at plus 35, but we are opening our voltage-gated potassium channels because their gate opens at plus 35. So now what's happening? Okay, so think about where potassium is. Potassium, we've got a lot more potassium inside the cell. Plus, now our cell is positive, right? So our positive charges want to move away from positive and into the more negative space. And we have way more potassium inside the cell. So if we're going down our gradient, we're going to do what's called potassium efflux, leaving the cell. Efflux means leaving the cell. So we are going out of the cell, both driven by our concentration gradient and the fact that our cell is now positive. So positive charges will leave and go to the more negative side. This is called repolarization. And again, that makes sense because now we're going all the way down, back more negative again. We're repolarizing down to those negative charges. And we overshoot a little bit. We get all the way down to -90 here. All right, and -90 is right about where our potassium equilibrium is. So that's where like the equal concentrations of potassium on one side or the other. And it's also where we close our gate. This -90 is known as hyperpolarization, where we've gone below our action potential. And it's also the point at which we can have, with enough signal, we could have another action potential. So up until we reach the hyperpolarization, going below the resting potential, this is what's called the absolute refractory period. And this just means we cannot do another action potential. Even if we wanted to, whenever we're above our -70, we can't like re-reach the threshold, right? Like there's no way to do it. We're already above threshold. So we have to get all the way down below. But then at this point, this relative refractory period, if we have enough signals to jack us back up from -90 all the way to -55, we could do that. So it could, in theory, go like bloop and go back up. Now, we need a lot of signal for that to happen, so we're often doing very high stimulus signaling if we're having an action potential form in that relative refractory period. But assuming that doesn't happen, then we just, once we close our potassium gates, this remaining component here is just getting us back to our resting potential, which is happening through all of the mechanisms here that we've talked about earlier. So our KEG ATPase, our leak channels, they're all helping to kind of reestablish that equilibrium. Because remember, when we're at -90, we're at potassium equilibrium. So there's probably not going to be much movement in those leak channels for potassium. So our sodium leak channels will leak sodium into the cell up until we reach around -70, and then we'll reestablish our equilibrium with gentle movement of the ions and the active pump. All right, I know this looks like a lot, but this is everything that's happening in the slice of the axon that is experiencing the action potential. Now remember, these axons are long, and these are very localized changes in charge. So we're usually having an action potential in just a section, a small section of the axon, and then that action potential kind of triggers the threshold to hit on the next part of the axon, and so it kind of moves down the axon as we go. So now let's return to our myelin sheath for just a second and see why the myelin can help us make that transition of action potentials down our axon faster. All right, so imagine everything we just did with our action potential happening here at the axon hillock. So we depolarize and we're sending that signal down. And then without this myelin, eventually that gradient change is going to dissipate, right? We're going to reach equilibrium with our pumps and our leak channels before it has a chance to get to that -55. And so like a lot of things that aren't insulated, that charge, that energy is going to kind of peter out. But by having these myelin sheaths, these are areas where we're not getting any concentration change. We're not moving ions around because we're kind of wrapping up this axon. And because there's no ion exchange happening where the myelin sheath is, that concentration change inside the axon is going to go all the way down until it can next move its ions. And again, that's the nodes of Ranvier. So what we have is an action potential, and then that causes a gradient change, and there's nothing that can be done because we can't be doing any ion exchanges with these myelin sheaths. So we have to move to the next small area, do an action potential then, and then scoot, next action potential, scoot, next action potential. And so what we're doing is, rather than doing action potential, action potential, action potential, action potential, like hundreds of them, we're only doing, relatively speaking, a few. And so we're maintaining that signal rather than letting it dissipate and reach equilibrium before we reach the synapse. And this conduction of kind of jumping from node to node rather than having them be this like continual gradient of action potentials is called saltatory conduction. Conduction meaning moving a signal, transferring a signal, and saltatory meaning jumping. Another interesting physics connection is how conduction versus resistance works. If you remember from your 4C topics, the equation for resistance is rho L over A. Rho is just the resistivity of the material, L is length, and A is cross-sectional area, right? So if we want something with low resistance, we want something with relatively less length and relatively more area, and that's going to decrease our resistance and therefore increase our conductance, right? We'll be able to move forward. So what we're essentially doing is having the length just be at these nodes of Ranvier, even though our axons are really long. And we're kind of keeping the area to a reasonable size. If we didn't have myelin, our neurons would have to be super thick in order to compensate for their length and have that decreased resistance through our cross-sectional area. A cool visual for just how important myelin is that our spinal cord's about, the neurons in our spinal cord are about the length of our finger, maybe a little thicker. And if they did not have myelin, in order to decrease resistance, we would have to increase the area to the point that our spinal cord would need to be like the thickness of like a birch tree or a telephone pole. Like it would have to be so thick in order to have the speed of transmission we need to get our signaling to our brain and then back to our body. So luckily for us, we're not the thickness of tree trunks in our spinal cord because we have awesome myelin insulation, which allows for that faster movement, that faster propagation. Again, if that doesn't help too much for visualization, just know myelin helps increase conduction by having us only have action potentials in small areas of the axon. Okay, ready to go back to our practice question with all of our channels described and identified? Let's go ahead and do it. The resting membrane potential of a neuron depends on, okay, we talked about three things, right? We have our ATPase, our sodium-potassium ATPase, and we have our sodium leak channel and our potassium leak channel. So the only thing that's ever active for our neurons function is our ATPase, right? So that is contributing to the resting membrane potential, and our leak channels are very passive leak channels that are always open. So the opening of voltage-gated sodium channels, that would be true if they had asked us about depolarization or the start of the action potential, like which of the following is required to start an action potential, or which of the following needs to be opened in order to start an action potential. That would be a really good answer for one. But voltage-gated sodium channels are closed during resting potential and they are not involved in maintaining it. So we cannot pick one, and on a Roman numeral question, as soon as you eliminate a Roman numeral, you eliminate all answers in that Roman numeral. Oh, look at that. We get down to C. So this is one of my favorite tricks with Roman numerals. Go Roman numeral by Roman numeral and see if you can eliminate one. Because if you can eliminate one, you can eliminate all answer choice options that include that. And sometimes that's all you need. We didn't even need to read 2 and 3. We just needed to eliminate one. But of course, we can do so. The active transport of sodium and potassium ion, that's our ATPase, and passive diffusion through potassium leak channels. Absolutely. I meant to write potassium leak channels, not sodium leak channels twice. So yes, our potassium leak channels are absolutely part of our resting membrane potential. They could have also written sodium here, and that could have also been a good answer, okay? Or they could have said the passive diffusion of leak channels for sodium, and then all three would have been correct. So several different ways to ask this question. They do love this question. They want you to understand the resting potential of a neuron. And now you do. We went over how to understand the parts of a neuron and how the resting membrane potential is maintained in a neuron and how the action potential is formed and propagated. I hope this video is helpful for you, and as always, feel free to share it with your MCAT community because we could all use a little help. I'll see you in the next video, and happy studying.